Bismillahirrahmanirrahim. Welcome back, friends. Welcome to another video sessions from Biology. In this video lecture, we will talk about the pathogenesis of viruses. Let me write. Pathogenesis of virus. Now, what does pathogenesis of virus means? Pathogenesis of virus means that once the virus enter, then how it can cause infection, or we can say that how it can cause disease inside the human. How the viruses can cause infection or how the viruses can cause disease inside the human. Fine. So it is called virus pathogenesis. Virus pathogenesis is actually a multi-step process. It contains three important steps. Once these three important processes or steps are done, then infection or disease may be occurred. How? Let me explain these three terms in a detail. First of all, the first steps in this case we call that attachment. Or adsorption. Now in this case what happens? The virus will attach on the host receptor. Once they attach on the host cell receptors, then the virus will be internalized. Or the virus, once they attach to host cell receptors, then there will be a bond formed. These bonds are called covalent interaction, or we call that covalent bond. How? Let me explain. Let's suppose this is a virus. Let's suppose. And in this case, the virus is what? The virus is a naked virus. Let's suppose. Naked virus means that they don't have envelope. These are the spikes protein arises, which are responsible for the attachment. And this green one represents the cell membrane or the host cell membrane. In this case, the host cell membrane also the eukaryotes and prokaryotes. Now what happened? These are the host cell receptor. So the virus will sit here, and what happened once the virus sit here? There are let me draw the genetic material here. The genetic material will be the RNA, DNA, or single-stranded RNA, single-stranded DNA. Any kind of the genetic material will be present. Fine. In this case, let's suppose they have double-stranded DNA or RNA. Now, once they attach here, so here the interaction will occur, or the bonding formation will occur, and we call that covalent bond, or we call it covalent interaction. Here, covalent interaction. Covalent interaction will occur. Now, once the covalent bond are formed, so the virus will completely attached. This is the first steps. They will completely attach, or we can say they are adsorbed. Once they are attached, then a second steps will occur, and we call that internalization. This is the second one, internalization. So there are two ways through which the viruses are internalized. One is cell-mediated endocytosis. In short form, we write CME. C means cell, M means mediated, and E means endocytosis. Cell-mediated endocytosis. And the second is membrane fusion. Now, what happened in cell-mediated endocytosis? In cell-mediated endocytosis, the virus will attach on the host cell receptor, and then they will be engulfed. Means they will take some portion of the host cell membrane, and then once they will engulf, so endosomes will form. how it will be look like. Let me draw. Let's suppose in this case the virus are now internalized. So how it will be look like? It will be look like just like in this form. Here in this area the viruses are now attached and viruses are pulled inside the host cell. These are the spikes protein and this is the virus. Fine. After some times the virus will fully internalized. This is the host cell membrane, and after sometimes what happen, these are the, so the virus will take some portion. Now these are the naked viruses, fine. So the naked viruses will be internalized through cell mediated endocytosis. So this is the naked virus, it will be internalized through cell mediated endocytosis, and it take some portion of the cell membrane whenever it internalized. So it takes some portion of the cell membrane, means this, the virus will completely engulfed. We can also say that. So now this is the virus which are internalized, and we called it endosomes, or when the cell internalized we call it endocytosis, and this vesicle are formed, we called it endosomes. What we called it? We called it endosome. Endosomes are formed. Fine. So here are the genetic material, DNA or RNA, any kind of the genetic material here. Fine. Now once it is internalized inside the host cell, then what happen, they will release out this capsid, and this vesicle means this membrane portion of the host cell will all the membrane portion, and this spikes area or this capsid area will remain outside, and means these area will be start degrading, and once this area will start degrading, then the genetic material will released out, and then transcription translation process will be occur. We'll be talking later, but now this is cell mediated endocytosis, and how the membrane fusion will be occur in this case. Now in the second one is the membrane fusion. So how the membrane fusion will be look like in this case? Now envelope virus will be internalized through a membrane fusion. Fine. When the cell have when the virus have envelope, so they will be internalized through membrane fusion. So how? Let me draw here. Let's suppose this is again a host cell membrane. Let's suppose this is a virus. The virus will be surrounded by a layer, outer layer, and we call that envelope, and inside this capsid there are the genetic material, any kind of the genetic material. Now from outside the spikes there are the from outside the envelope there are the spikes arises, which are responsible for the attachment. So what happened in this case? They will also attach to the host cell receptor. Once they attach, again the covalent interaction will occur, and after the covalent interaction they will be internalized. So how they will be internalized? They will be internalized through membrane fusion. How the membrane fusion will be happening and how their structure will be look like, let me draw. In this case, the envelope area, let's suppose what happened. The envelope area, this is the virus. Fine. So now these are the envelope area of the virus. So what happened in this case? The virus will leave their envelope area, means they will fuse their envelope area with the host membrane, and just they will release what? They will just release their capsid area. So just capsid area will be released, having the genetic material also. So only the capsid area will be internalized. So this is the membrane fusion. Fine. So they will be, so the envelope virus will be internalized just like that. Fine. Now this is the second, and third we have the last, we can say last we have the unpacking of the genetic material. Unpacking of genetic material. What happened in this case? Now let's suppose the virus is fully internalized through cell mediated or through a membrane fusion. The virus is not internalized. Once the virus will internalized, then they will release the genetic material. So let's suppose this red one represents the genetic material of the viruses. Once the genetic material are internalized, not only the genetic material, the proteins will also internalize, means the capsid area will also internalize. Some viruses means some of the capsid will be internalized on the host cell on the host cell membrane, and some virus will be means some viruses will go inside the nucleus, and when they are going inside the nucleus, before they going inside the nucleus, they will leave the capsid area, and just the DNA portion will be internalized. Fine. So here let's suppose the virus will now present inside the nucleus. So the capsid area will remain outside, and the virus just inject their genome. Fine. And the protein portion will also remain outside the nucleus. Then what happen? Inside the nucleus there are the polymerase enzyme, and every proteins are present. So this genomes is used as a template, and then they produce the early messenger RNA. Early mRNA. And after this early mRNA means the transcription process occur, which can produce the messenger RNA. Not only erase this one, not early mRNA, means that the transcription process will occur, they will just produce messenger RNA. Once they produce the messenger RNA, then from messenger RNA translation process will occur, by which they can produce the proteins. So now let me draw the proteins here. These are the proteins. These are the structural proteins. Fine. Now these are the structural proteins. And these structural proteins are used to make the capsid, and around the inside the capsid there are the genetic material are also present. So means the virus. So what happened? Let me draw here. This is the capsid portion. Now the capsid portion will formed, and inside the capsid there are the genetic material genomes of the virus, DNA, RNA, or any kind of genome. But these viruses are just, but here just capsid are formed. Then after that what happened? They will go, they will release out from the nucleus, and they will also take some portion of the nucleus. From the nucleus, then they will reach to the endoplasmic reticulum. So after that they will reach to the endoplasmic reticulum. Let me draw the endoplasmic. So endoplasmic reticulum will be look like in this form. If you don't know the structure, so go to you Google and just write endoplasmic reticulum and watch the picture that how the endoplasmic reticulum will be look like. In the endoplasmic reticulum there are the proteins, means the receptors are formed. So these receptors are actually glycoprotein. So that's why the virus should should pass from this endoplasmic reticulum. Once they will pass from the endoplasmic reticulum, so they will receive the glycoprotein, and once they receive the glycoproteins, so the virus will now completely formed, but still it is not mature. So what happened? The virus will be look like just like in this form. Now here are they also take some envelope, means they also take the envelope, and here are the spikes. If let's suppose if the virus is envelope virus, fine, so they will take the envelope. But if the virus is naked, so the virus just take here the receptor. Keep this thing in your mind. When the virus are the naked virus, so they will just take the receptor here, means the glycoprotein just they will take. But if the virus is envelope, so they will take both receptor and envelope portion from that endoplasmic reticulum, and after that they will go to the Golgi bodies. Also, if you don't know the structure of the Golgi body, so also go to the Google and search the Golgi body, that how it will be look like. Now they will pass from the Golgi bodies, and from the Golgi body the virus will be matured. So we get the mature virus. We get a mature virus. This mature virus will now ready to. Now there is one other important thing that I want to tell you. Now listen. When the virus are envelope virus, so they will be released from the cell through a membrane budding process. Means they will take some portion of the membrane also. Or if the virus are naked virus, let's suppose if the virus are naked virus, then what happen in naked case? Naked virus will cause the cell lysis, or they will burst the cell and then they will release. But the envelope virus will not causing the cell killing or the cell lysis. They will be released out from the cell through a membrane budding, and they will be released. Naked virus will be released through cell lysis. So the release phenomena are also different in case of envelope and in case of naked viruses. Fine. So that's how the viruses can cause. That's all about the pathogenesis of the viruses. I hope you understand. If you like the video, please hit the like button, share this video, and write a question in a comment if you have question. And I will try my best to give you answer as soon as possible. And share this video with your friends. And also subscribe the channel to get more interesting video lecture like that. Thank you so much.